And we're going to go through the slides. What I'm gonna tell you about is about two different larger groups of freshwater mussels. And they are distinguished from each other by their sort of natural life history traits. So we have basically, there's a group called the Unionids, and they're remarkably interesting because in order to complete their life cycle, they have to spend some of their time occupying the bodies of another organism. Not only another organism, but an organism from a completely different phylum of organisms. And so here what you can see in your slides, if you're following along, is that we're showing you that this type of freshwater mussel releases something called glaucidia into the environment that then have to spend time on a fish. They travel around on the fish and become juveniles eventually, and then of course the juveniles become adults and the cycle is complete. This means essentially that they need to parasitize a vertebrate species in order to be able to survive and to breed effectively. It's kind of neat. So we're going to kind of tease this apart a little bit and look at some of the different ways that this life cycle can be completed. The Unionid group have what are called glaucidia larvae, and this is different from the veliger larvae. So just kind of put veliger out of your minds for a second. We're only going to be talking about glaucidia larvae in this mini lecture. You are going to be responsible for learning about veliger larvae in the same way that I demonstrate here. So, glaucidia larvae. I'm kind of picking the really cool one compared to the veligers, but basically glaucidia are little sort of larvae that travel around and are specially adapted to travel around in a host species like the fish. So they basically need all sorts of things like ways of getting around, ways of getting to the host, ways of staying in the host, and very importantly, ways of releasing from the host when it's time. And that's basically what a glaucidia larvae is. So what you can see in this slide then is all sorts of different types. Some of them are very big, others are very small. And in this particular case, this group of freshwater mussels, the glaucidia are parasitizing the gills of the host fish. So here in this slide, we have gill tissue where you can see 4 glaucidia that have attached themselves to the actual tissue of the fish. They do this because they kind of have these little sort of pinchers. Their whole body basically looks like Pac-Man, where they open and close and hook onto the gill tissue of the host species. So one of the things that you may be asking yourself, hopefully you are, is how do the glaucidia get from the adult freshwater mussel to the host species? And this is where, this is the kind of the cool part. There are a bunch of strategies. One of the strategies is to use something called a conglutinate. And basically what it means is that the freshwater mussel, the species that uses the conglutinates, packages up the glaucidia in a kind of bundle and tries to make or makes that bundle very, very attractive to the fish host species. In fact, so attractive that a remarkable thing has evolved. The conglutinates look identical to the natural prey item of that host species. So basically, if a host species, a big fish, eats little fish, there are conglutinates out there that look like little fish. And this is what is shown here in this image. These are not little fish. These are conglutinates of the freshwater mussel, so tissue of the freshwater mussel. It kind of sends it out on like a long spindle into the environment and waves it around. It looks like a fish, as you can see, but it isn't. It's basically just a sac that is full of glaucidia. The idea being then that the fish comes along, thinks that it's prey, grabs it, busts it open because it's kind of a delicate structure, and then gets infested with all sorts of Glaucidia. Here's another example from another species, and instead of looking like little fish, this actually looks like a tiny little invertebrate, basically like a little Daphnia larvae that is eaten by a particular host fish species. So it looks identical to those larvae that it would be going after, but in fact it's just a sac full of Glaucidia that's made by the freshwater mussel in order to transfer the tissue. It's kind of neat. So here's a kind of image that shows the whole system. Here we have the host fish, the fish that's going to be parasitized by the Glaucidia. Here in the background we have the mussel, so it's rather large compared to the size of the fish. And just over top of the fish we have two conglutinates, which are these prey mimics that basically serve as a means of luring in the host fish and then infesting it with the Glaucidia into its gill tissue. So let's see this in action. I'm going to play this twice because it's rather fast, but it's very cool. This is a video, and I'll post separately the videos because I know that the screen is a little over or underexposed. Follow along in the slides that we've posted those so that you can see if there are words that are unclear in this video. At least you'll be able to read them on the PDFs. But I'll play this video. What we have here is a fish, a host fish, and right here is a conglutinate. It's just kind of acting like a little lure, attracting the fish. And I'll play the video twice. So that was once. What you saw there was the fish responding to what it thinks is prey, basically disrupting the tissue of the conglutinate with its mouth such that the Glaucidia are released. And if you watch this video on your own monitors using the links that I provide, you'll actually see some of the Glaucidia floating around the head of the fish. When the fish realizes that this is not food, it spits it out again, but the damage has already been done. So I'll play it one more time. It's kind of neat, right? It gets weirder. Another way of transferring the Glaucidia directly to the host species is to physically hold on to the face of the host fish. It sounds a little bit rough, but what you have is some species have evolved these special tooth-like structures. Now they're not teeth, they don't have dentine or enamel or anything that defines a tooth, but they do serve sort of a very similar function in that they're meant to grab and hold on. And so little spikes on ridges is kind of a standard way of overcoming a problem of needing to hold on to something. And what we show you here are a couple of examples, different species that have evolved these tooth-like structures. Instead of making conglutinates to kind of attract, what these species do is they attract the prey in another way, either by having sort of some flashy tissue that attracts attention, or having what's called a lure, and we'll get to that a little bit later. What happens is, once the mussel has detected the presence or the interest of a host species, what it will do is it will close on top of the face of the fish. Now, not to kill it, that would be detrimental to its cause, but rather just to hold it in place while it then releases Glaucidia into the environment around the fish's face so that the Glaucidia can infiltrate the gills. And we have a video of that, so I'll show you that. So what was going on there is that as the fish became interested, the freshwater mussel detected its presence, closed on its face, and then release the Glaucidia into the environment right around the fish. So play that again if you want, just rewind and see that again. And you'll see that it's really quite violent looking. Researchers have discovered something very cool about how to collect Glaucidia from this type of freshwater mussel. Basically, it turns out that you can't just stick a tube into the freshwater mussel and hope that the freshwater mussel is going to release the Glaucidia. Freshwater mussel is a little smarter than that and doesn't waste its energy on something that isn't a potential host. So the researchers weren't able to actually get any of the Glaucidia that they needed in order to conduct their study until somebody figured out, it's kind of weird, somebody figured out that if you take the head of a fish and stick it on the end of the tube, then you fool the freshwater mussel into thinking that it is a viable host species, and it does close down on its face, and it does release the Glaucidia. So check out this video. So here we have a fish head where there's a tube that's inserted into it, a freshwater mussel who's now attached to the face, and if you watch this on your own, on the link that I provide, you'll actually see Glaucidia getting sucked up the tube, where if you had just used the tube, the freshwater mussel would not have released the Glaucidia into the environment. So figuring out how to attract the fish. So now we've kind of gone backwards. We've shown you how the mussels get around by using the Glaucidia. We showed you how the mussels invade the host gill tissue, so how they actually get the Glaucidia into the gill tissue. And now we're going to look at how they attract the fish in the first place, because mussels are sedentary for most of their life cycle, but fish are not. And they do this by kind of showing them shiny things that might be of interest. So the conglutinates, for example, where they actually mimic the prey items. Another way that you can do it is to have a lure, which is a little different than a conglutinate, because a conglutinate is kind of an all-in-one package, where the package looks like the prey item. A lure is simply that. It doesn't actually have the Glaucidia inside, but it's there as kind of a shiny thing to attract the fish. And of course, we can have different types of lures. Some of them are very simple, so just wavy things like a flag, but others are actually very complex. And I'll show you some of the diversity. Here in this slide, what we're showing you is a lure that's here, and it's very difficult to see, but it's a rather simple lure. I'll show you a zoom in just a second. Here we go, a little bit clearer to see this GIF here, especially the one here on the left-hand side. This is what a lure is, and if you can take a look, you can see the shell of the freshwater mussel that's here, and it's open slightly, and it's extended out this lure. And this lure looks surprisingly like a fish, right? And the way that it's pulsing, too, suggests the movements of a fish. But in fact, it's just part of the mantle tissue of the freshwater mussel that's used to catch the eye of a prey item, or of a host species that thinks that that's prey. It's a way of attracting that host fish to it.